Good morning, my name's James Stanger. This video is the first in a series I'm going to make on publications talking about euploid embryos. Number is my intention 001. It's going to be a lot. Okay, so this is a new series of videos I'm making concentrating only on studies using euploid embryos. They explore the basic embryology, embryo behavior, and implantation associated with true euploid embryos. And in so doing, it excludes the impact of aneuploid embryos, especially on pregnancy rates. I will discuss several recent publications that explore what conditions or observations are related to euploidy and what may not. Just remember that euploidy is currently determined by a PGT-A process that may induce some flaws. Non-invasive PGT-A may or may not provide a different outcome. So, at the start of these videos, I'm going to present two views on the topic. One will be for IVF staff and one will be for any IVF clients who may be watching this video. So to staff, you make the point that euploid embryos are determined after you're doing PGT biopsy and genetic analysis. They represent viable embryos for transfer and storage. They may either be euploid or minimally mosaic embryos. They are identified by the biopsy of trophectoderm and not the inner cell mass. They may not truly represent the future health of the baby, but they do have a 50% lower incidence of miscarriage. Since after transfer they are most likely to result in a live birth, many studies are now concentrating only on euploid embryos. What can they tell us about implantation and IVF? Is this becoming a new standard? If you're a client of IVF, the important piece you need to have pointed out to you is one, that about 40% of all embryos have the correct number of chromosomes. This is called being euploid. This can only be determined currently after biopsying the embryo, setting it off for genetic analysis, freezing the embryo by vitrification, thawing them out and transfer it. It involves cost and delays. Euploid embryos have a lower chance of pregnancy and in some studies a shorter time to pregnancy. Some embryos labeled as not euploid, that is called aneuploidy, may overestimate the likelihood of the inner cell mass and the baby being abnormal. Therefore, some embryos will be discarded, which otherwise may give rise to healthy children. Some embryos are a mixture of euploid and aneuploid cells, called mosaic, and the degree of mixture can reflect the health of the future child and the likelihood of pregnancy. And this, of course, is the subject of future videos. I'm going to discuss 4 papers that have been published recently: embryos not showing pronuclei by static observations, live imaging of late-stage pre-implantation embryos revealed de novo mitotic errors, euploidy rates within blastocysts obtained from different size cohorts and age distribution, and the inner cell mass grade and early blastulation are associated with pregnancy outcome in euploid embryos. And just to be mischievous, I'm throwing in one from 2021 on morphology-based selection. We shouldn't really be in this video, but I wanted to keep it anyway. Okay, the first one. This is an article published in Human Reproduction. It is not open access and was directed towards, as the study question points out, is the non-pronuclear fertilization pattern as assessed by a single static observation during the 16 to 18-hour post-insemination window compatible with embryo developmental competence? And the answer is a few zygotes, that is a one-cell embryo, show pronuclei solely outside the static range of 16 to 18 hours and can develop into blastocysts. But while they also make a point, which isn't the subject of this video, that some rare cases of cleavage without pronuclear formation always result in early development arrest or degeneration. So this is an embryo. The day after we've added sperm or injected it with the sperm. And what we're talking about here are these 2 pronuclei. These are the chromosomes from the egg and the sperm. The larger one is usually from the sperm, the paternal pronucleus, and the smaller one is from the egg. There's a pole body up till it's been fertilized, and this is zona pellucida. You can tell this is IVF because there's sperm bound to the zona pellucida. And this is one of 2 critical tables, so it's pretty hard to comprehend. Basically, they're looking at the embryos that started to appear first. This is the time pronuclei first appear, under 16 hours, between 16 and 18 hours, and over 18 hours. And then the time these pronuclei fade, I'll explain this in a minute, against 20 hours, which is a fairly long one, the embryos that fade sooner, do they have a better chance? And basically, if I just go back and look at these pictures just to explain it, this is the zygote. This is one of the first pictures you get from time-lapse imagery, where you can see the fertilized egg, still a single cell, a polar body on top, which hadn't yet been, and there's no sign of pronuclei inside. But within several hours, that polar body, there's now two, one from the original one being extruded from the second mitotic division. And you can see these two pronucleus, two swelling of chromosomes just in the center of the egg. And bit by bit, they get larger, they come closer together. And of course, with time-lapse imagery, you can't always control the orientation which you're looking through the egg. So I haven't on purpose, but I have chosen one where it's slightly oblique. But you end up round about 16, 18 hours, these two pronuclei are butting to each other very closely together. Then all of a sudden they disappear, and within 1 or 2 hours, the embryo divides. The initial division is quite ugly, but they smooth up very quickly within a few hours. So we're talking about the time these two pronuclei appear and the time they disappear. So going back to that slide again, the interesting observation is that the chance of this is a chance of a 2PN zygote forming a blastocyst. And the first thing is, if you look at maternal age, the ability to form blastocysts is highly age dependent. And the second thing is comparing to sequential media, which has always been the default media, that is a media for early cleavage and a media for later cleavage, using continuous media, which is suitable for time-lapse incubators, there is a significant improvement in the blastocyst formation rate. In other words, using continuous media and all the time-lapse environment produces more blastocysts. But comparing embryos where the pronuclei first appeared under 16 hours, you can see that pronuclei that appeared between 16 and 18 hours was not significantly any closer, but embryos that appeared after 18 hours had a significantly lower chance of forming a blastocyst. So basically saying 16 hours is a good time, but where the embryo takes longer to form pronuclei, 16 hours or more than 18 hours, odds ratio 0.25, highly significant. So an embryo is taking more than 18 hours to form pronuclei, that's bad. Conversely, if you look at when the embryo's pronuclei disappeared, the fading, compared to 20 hours, embryos that fade between 18 and 20 hours have a highly significantly higher increase of forming a blastocyst. But if they fade too quickly, 16 to 18 or earlier than 16, there is no significant improvement. So the summary is, this is for blastocysts. This is not for euploid, right? That's the next slide. So this is just to form a blastocyst. After adjusting for maternal age and the use of continuous versus sequential media, the time for appearance greater than more than 18 hours post-insemination was associated with significantly lower blastulation rates compared to less than 16 hours. And the time to fade of 18 to 20 hours was significantly higher blastocyst rate compared to fading more than 20 hours. That is, a slow pronuclear development is associated with poor blastocyst development. Fast pronuclear completion is associated with better blastocyst development. In other words, the pronuclei should appear, do their business, get organized, and then fade and get onto the first cleavage very quickly. And any delay in the formation or the time taken to complete the pronuclear processes is a poor indicator for blastocysts. If we went back and do the same thing just for euploid blastocysts, which is one of the purposes of the study, the authors point at this stage that the time the embryo pronuclei first appear doesn't become that critical. But compared to more than 20 hours, that's a long time to fade. The optimum time to form a euploid blastocyst is 18 to 20 hours. So in other words, if you're doing a static observation and you're trying to accurately predict the chance of a euploid embryo, 18 to 20 hours post-fertilization with the ICSI would be the critical time, and you can't, if you're outside of that, you won't pick it up. So after adjusting for maternal age, only the time fading for pronuclear to fade 18 to 20 hours showed significantly higher euploidy rates than other embryos. Okay, and I'm not sure whether I can really take this from the paper because it is not open access, but it's such a fantastic graph. These are the times for euploid blastocysts and non-euploid blastocysts, and you can see here this is the time for fading and this is the time for appearance. There's a very narrow window when they appear, but the time for fading is quite broad. But optimally, it's round about this time here. So the rate at which pronuclei develop and disappear is loosely related to blastocyst development and to a lesser extent euploidy rates. Prior to their appearance, the time is spent unraveling of the chromosomes and for the male DNA replacement of protamines with histones. There's obviously a lot more going on, which isn't the subject of this talk. For the pronuclear stage is a period where the embryo is checking the DNA and starting to activate the embryonic genome. Damaged sperm DNA and slow oocyte activation by phospholipase C are two recently identified factors that may contribute to delays. There seems to be a strict time for pronuclear appearance and a larger range for pronuclear disappearance, which is related to euploidy. The second paper is in Fertility and Sterility Reports. It's an open access article. And even though the conclusion here is a little bit complicated, the next graph is the interesting one. So this is a graph from, I believe, America. And they wanted to investigate whether the distribution of euploid blastocysts within cohorts can be predicted. Okay, the cohorts here are the age and the number of blastocysts available to be biopsied. And they concluded the probability distribution structure of euploids within discrete cohort sizes is binomial with all age groups and nearly all cohort sizes. A bit hard to get your head around. And then just make it more confusing, they say the probability distribution ranges identified represent a more accurate frame of reference that describes and predicts the number of euploid blastocysts most likely to be present with an individual cohort before biopsy compared with estimates from simple age group estimates. In other words, they're saying the age does impact it, but also the number of blastocysts available was also important. These results offer new approaches for counselling and clinical decision making. The point here is that before you encourage the client to proceed doing blastocyst biopsy and PGT, which needs huge cost, and a delay in treatment, you should have some idea of the likelihood of how many euploid blastocysts you're likely to get. And in this study, they're looking both at the age distribution and the number of blastocysts available for biopsy. And this is the graph. And it actually looks complicated, but it's actually very interesting. It's a really good graph, actually. So I've summarized some of their other tables to suggest that the proportion of euploid blastocysts per blastocyst is determined by age. Overall, they had 61%. And at this point in time, I need to stop and go back and say that most studies report only about a 40% euploidy rate for blastocyst biopsied. But in this group, where they include mosaics up to 20 to 40%, then that adds another 20% of euploidy rate. So 61%, 41 would be true euploids, and the other 20% would be low threshold mosaics. If you look at the age distribution, this is the important one. For people under 30, and there's not that many people having IVF under 30 these days, there's 3 chances in 4 of an embryo being euploid. And it falls 68, 57, 44, even 44 is 50%. If you're under 40, you're still the reasonably good odds if you have lots of eggs. But over 41, at the euploidy rate per blastocyst biopsy really falls away quickly. So not only are there fewer eggs, but there's also fewer of those will form a euploid blastocyst. That is, the impact of aneuploidy is starting to make real impacts somewhere over 40. If you go back and look at the table, this is broken into age groups. I think these are the start age groups they use. And this is the number of cohorts. We can see this is where they have one blastocyst for biopsy, 2, 3, 4, 5 blastocysts for biopsy. And there are cases of 10 or more blastocysts available for biopsy. That's a huge number, and a huge amount of work. The embryologist must be exhausted. That's a lot of work for one case. So this is a group, this is a box of whistle cleaning, and you want to look at the mean, which is the dark line. And you can basically say if you have one blastocyst and you're under 30, you have a fairly good odds of getting that being euploid. But as you go on to more basically, it starts to fall away a little bit. But if you, for instance, look at 6 blastocysts for biopsy, on average, the mean here is roughly 5. So even with 6 blastocysts to biopsy, you may reasonably expect to end up with 4 or 5 blastocysts. And look at the number 10. There's rare cases where these are almost PCO cases. Where you're having 10 blastocysts to biopsy, even then you would expect to have 8 being euploid. And I was thinking that this would be a fantastic group to look at the cumulative live birth. When you start putting back lots and lots of euploid blastocysts, what is the cumulative chance of pregnancy? And that gives you some idea of what other things are impacting. But when you go back and look at the cohort 30 to 34, which is a more common age group, the relationship is still the same. That is the binomial, but the numbers are slightly less. But even with 10 blastocyst biopsy, you're still going to end up with 7, maybe with 6 euploid average every 10 for the 35 to 37, 38 to 40. You're getting down to around 4 or 5 blastocysts, but that's still a lot. But if you're getting down to 1 or 2 embryos in the 38 to 40 group, then there's chance you'll actually not have any. But crossing the Rubicon at 40 years old, it really becomes apparent that this is when you really start to have trouble. Even though you can have cases, like these are 41 to 42-year-old women having 10 or more blastocysts to biopsy. In that case, you may expect to get 2 euploid. That's enough for a transfer, 2 transfers. But if you're 41 to 42 and you only have 2 or 3 blastocysts, there is a significant chance you'll end up with none. And doing PGT for people over 43, where you're trying to work out which one's euploid, you're almost asking to have your transfer cancelled. And this is a group that I think needs more counselling. Some people may prefer just to put the embryos back and run the risk of a slightly higher miscarriage rate rather than just walking away. So the results: a probability that a blastocyst euploid decreases with maternal age, but increases with cohort size into 6 stratified age groups. And there are a lot of people now using AI to use both the age and the number of blastocysts available, the number of eggs collected, to try and make predictions. But this is kind of the data that's presented on. I actually think that was a really good paper. It took a while to get around it. This one, this is basically the same one. And they're basically saying that my comment is the number of euploid blastocysts is largely linear to the blastocyst cohort size. It's largely age-related, with older women having fewer euploid embryos per blastocyst than younger women. So age impacts both on the number of oocytes and blastocysts and embryos, but fewer euploid embryos per fewer blastocysts. Older women opting for PGT-A are increasingly less likely to have viable embryos for transfer, but younger women may reasonably expect that 2 in every 3 blastocysts biopsied will be suitable for transfer. Okay. The next paper I'd like to talk about appeared in Nature Biotechnology, and it talks a little bit about the accuracy of PGT-A. So these people, they used a new imaging technique that allowed scientists at the University of Cambridge to observe that some genetic errors appear late in embryo development than previously thought and were present only in cells destined to become part of the placenta, as the trophectoderm. This means that some embryos flagged as abnormal may still be viable and could influence how clinics select embryos. Now, I have to say, when you read the paper, they're not looking very much at the number of cells in the mitotic error rate in the endocell mass. This is only in the trophectoderm. They discovered that some chromosomal abnormalities arise after cells that would normally become placenta, that is, cells that have always differentiated become trophectoderm, have already diverged from cells that eventually become the body or the endocell mass. If the embryo were to develop into biopsy cells, were taken to be representative of all the cells in the embryo. However, this research shows that embryos can have changes in these trophectodermal cells that may not be present in other parts of the embryo that go on to form the baby for fetus and baby. Therefore, we're giving a potential misleading picture of the embryo's overall viability. As a result, the studies suggest that some embryos may be discarded unnecessarily, potentially affecting patient outcomes and costs. So the authors made our findings challenge the current practice of trophectoderm biopsies for pre-implantation genetic testing for aneuploidy, a widely used clinical method to detect aneuploidy in human embryos. It is generally assumed that the aneuploidies detected originate from early mitotic or meiotic errors. However, we provide evidence using advanced chromosome tracking and live embryo imaging and segmentation technologies to show that de novo mutation errors can arise late in human pre-implantation development. They say that our data support the conclusion that these errors may be confined to the trophectoderm, leaving the inner embryo-fated cells unaffected. They also, I don't know why I included this also observed periodic cycles of blastocyst growth and collapse, which are accompanied by fluctuations in nuclear volume and I think the rates of mitotic errors. The current labeling method predominantly marks trophectoderm cells. As a result, it is not yet clear whether similar changes occur in cells of the inner cell mass at the stage. So they're making a comment that such embryos would normally be called mosaic, and as indicated before, many embryos defined as euploid may end up containing up to 40% mosaicism and still be transferred. And I guess what they're suggesting here is that there is this cutoff of roughly about 30-40% mosaicism will still give rise to healthy babies, but more than 40% mosaicism starts to give rise to a lower pregnancy rate. And they're suggesting that maybe some of these embryos at the very cusp of cutoff from mosaicism may in fact be an artifact of atypical mitotic errors in the trophectoderm, and which may not reflect the inner cell mass. And the only problem about this paper is it's implying that the inner cell mass is not affected by this, but there's no data to show that the inner cell mass cells don't have the same rate of late stage mitotic errors. And this one I just thrown in just to be mischievous. This was published in 2021. A bit out of date. I might actually go back and do a whole video on this. But they're basically just talking about rates of development and euploidy. But they're basically saying that the sex proportion was impacted by the day biopsy and trophectoderm morphology, and not by inner cell mass morphology in PGT cycles. So this wasn't an open access. This is only just from the abstract. Therefore, biopsying an embryo on day 5, based on good quality trophectoderm, which is what you need to biopsy, shifts the male proportion towards males if you're only doing biopsy on day 5 of good quality embryos. They make an interesting comment though. We read the results section of the paper. The sex proportion was impacted by the day of biopsy and trophectoderm morphology, but not by inner cell mass morphology in PGT-A cycles. Therefore, if you're biopsying really good quality embryos with good trophectoderm on day 5, and then you're only biopsying those, then you're going to shift the proportion towards males. Interesting, they noted that the morphology-based embryo selection for single embryo euploid transfers has favored the choice of XY embryos generating a 55 or 54.3 male proportionate transfer and 56 proportion of males for ongoing pregnancy. The models indicate a weak association between blastocyst morphology and gender. That is, there is a little bit of bias, but it's not very much. But on a similar sort of vein, this was the paper that led me to the other one. This was another study published in October in Reproductive Science, where looking at the inner cell mass grade and early blastulation, they argue that the embryo sex was known in 90% of transferred embryos, with 51% female and 49% males. That is, we're just putting back embryos based on quality, etc. Then by natural choice, 51% would be female, 49% would be male. But in 161 cases, 1 in 5, 20% of cases, where the embryo's gender was used to select the embryo for transfer, it resulted in only a very small shift. That is, you're using the gender to make the opinion, but you're still going to only go gender. You're only going to bias it to 52%, 47% female. In other words, it's really not, if you're still picking out the very best embryos, it's not really going to make a lot of difference on the sex ratio of the children. If you look at the data though, it's kind of interesting, where you're looking at male embryos. This is the chance of looking at the scores and the chance of pregnancy of euploid embryos. The male embryos, the day of vitrification was the most highly significant effect. The inner cell mass had a smaller p-value, but wasn't significant. So for male embryos, the day of vitrification was the key marker. But for female embryos, that wasn't so much the case. It was more the quality of the inner cell mass. So there is something funny happening here whereby between male and female embryos, the male embryos seem to be developing a little bit faster and with a slightly better trophectoderm, whereas the female embryos may not have a trophectoderm development at the same rate, but far better inner cell mass. And they had a table here which looking at the birth rates by sex, quality inner cell mass, and day of vitrification. And so if embryos frozen on day 5 with good quality inner cell mass, 6% were female, 61% were female, 46 or 56 for male, and for better quality inner cell mass, it's roughly the same. And even for day 6, the inner cell mass embryos give you a better outcome. So they found that both the day of vitrification and the quality of the inner cell mass of vitrification were significantly associated with odds of live birth in euploid embryos. In contrast, the quality of the trophectoderm of vitrification, the sex of the embryo, and whether the embryo was selected for transfer based on sex was not associated with live birth. So in conclusion, euploid embryos with the better ICM quality and those that reach the blastocyst stage earlier are associated with higher odds of live birth, suggesting that prioritizing embryos for transfer based on these parameters may provide patients with the best chance of pregnancy. And you may actually say that looking at all the algorithms, looking at all the different stage of embryo development, I wouldn't be surprised if the AI is picking out the quality, the size of the inner cell mass, and the time as being the ones that give the highest KIT score. Okay, so in summary, there seems to be a strict time for pronuclear appearance and a larger range for pre and disappearance and euploidy. The number of euploid blasts is generally linear in relation to the blastocyst cohort size. This is largely true for younger women, but older women are having fewer euploid embryos per blastocyst than younger women. Some authors have suggested that de novo mitotic errors can arise very late in human preimplantation development, and the conclusion is that these errors may be confined to the trophectoderm and may not affect the inner cell mass. As a result, some embryos may be being discarded during the late stage mitotic errors, while the inner cell mass and future fetus and baby are entirely normal. Therefore, biopsy on day 5 with a good trophectoderm shifts the sex population towards males. But if you really want the highest chance of pregnancy, another study suggested with euploid embryos, the better ICM quality and those that reach the blastocyst stage earlier were associated with higher odds of live birth, suggesting that prioritizing embryos for transfer by these parameters may provide patients with the best chance of live birth. And that, they say, is your lot for the week. Just as basically a disclaimer, my name's James Danger. I'm a member with 50 years experience. And I'm not a medical person, and this information is not medical advice, but it may help you in knowing what questions to ask about your treatment with your doctor, and maybe to help in understanding some of the advice you receive. So I'm just starting out, but if you'd like to promote your company email, this may be a good place to do it. If you like what I'm doing, click subscribe and or maybe become a sponsor. And if please, I always leave a comment and say, if you like it, click like. And just a word from my own sponsor, which is my own company, Third Aid. This is an online EQA and training program in IVF and andrology. It provides EQA schemes training in online for concentration, motility, morphology, sperm DNA fragmentation, halosperm, and vitality. And it does embryo grading for follicles, oocytes, zygotes, cleavage, and blastocysts, both static image and time lapse, and do embryo ranking. You can enroll as a trainee or as a professional. It can accommodate small clinics or large multi-lab clinics, and it can be used for training or competency or for accreditation. And that is the end. I'd like to thank you for your patience, for persevering. I understand IVF is a personal journey that involves the action of many, and it helps to understand all aspects of that journey going forward, both your own experience and the advice you give. I hope this information may be of value. If you like the information provided, please tell the clients about the service and please subscribe. And that, I'll say goodbye.